Hi everyone, this is Mr. Sinti and I want to talk to you a little bit about polymerase chain reaction. After we extract DNA, the next step in the workflow is to take the redwood DNA that we've extracted and use a technique called polymerase chain reaction. And simply stated, it's DNA replication in a test tube. And so if I can illustrate that a little bit, and let's say the DNA, I'm not going to draw it the helix like this, but let's say that the redwood DNA that we extract, the genomic redwood DNA, so it's gDNA. The genomic DNA of a redwood tree contains, like from one segment to another, it contains this gene, and it has from here to here is another gene. So this is gene 1, this is gene 2. There's thousands of genes along the redwood DNA. And what we're interested in is just this one area, this one segment, like let's say from here to here. And if you remember from a previous discussion that we've had, it's like what gene are we interested? What is the target gene that we want to make many copies of, or if you will, amplify? We're interested in a gene that is called GAPDH. It's the gene that produces the protein glyceraldehyde phosphate dehydrogenase. And so what's interesting about that is how is it that we can make many copies of that one area and exclude the other pieces of DNA? So basically in a test tube, how is it that we're going to be able to replicate just this area over and over and make millions of copies of this and not make millions of copies of this other area right here? Let's take a look at that. Well, the DNA that we put inside the test tube is going to be called template DNA, and so obviously this comes from the redwood tree. And so this is a molecule that contains the segment that we're interested to amplify, and that is the gene for GAPDH. And so what we're going to do is, in order to amplify just the area of the DNA, if I were to extend this and say, all right, here's DNA going in this direction and this direction, like this, but we're only interested in this one region right here, the template. How we're going to do that is in our little test tube, we're going to add primers. Now, primers are these little small segments around 10 nucleotides. They're called oligonucleotides for a few. These primers are going to attach to right in front of our gene over on one side and right on the other side of the other strand. And so when DNA polymerase, let me look ahead just a little bit, when DNA polymerase comes, which is the enzyme that replicates DNA, if you remember that, it can only attach new pieces of DNA to a growing 3' direction. And it can only attach a nucleotide if there's already a nucleotide present. So these just attach. They find the segment that is complementary, and then once they anneal, then the polymerase comes and goes like that and makes copies. And so it's only going to make copies in this one particular area, which is really cool. And so these primers are little pieces of DNA that we can order from a biotech company, and we just simply place the primers into a test tube with our redwood DNA that we've extracted, and it's unbelievable, but out of all the billions of nucleotides, these primers will swim around, and they're going to only attach just to the area that we want them to. It's unbelievable. This is how we can look at different, if we're interested in studying humans, this is how we can actually look at the differences between one person and another. We can look at whether or not a person is a carrier for a particular disease or not without going and sequencing the whole genome. We can just look at a target area. So it's unbelievable. So they're really specific and they attach to both sides of the gene. And this is a really cool picture too. This is a close-up. So it's a small piece of DNA. We need a forward primer and a reverse primer. They attach. And so just this area in between where they attach is the part that's going to be amplified. Now, in order to amplify the gene, you need some components. And so one of the things that you need is this enzyme DNA polymerase. It's going to be the one that replicates the molecule, and it's going to grab DNTPs and it's going to start to elongate the molecule. But it's like, well, what kind of enzyme could handle extreme temperature? If you recall, I may have mentioned this before in a different video, but I'll say it here. In order for us to open the molecule up without using DNA helicase, we need to use a tremendous amount of heat, about 95 degrees Celsius, to get the DNA to open up. Well, proteins don't like that kind of extreme temperature. The heat agitates the molecule, and what's holding it together and making it work properly is sort of just electrical bonds between the R groups of this protein. If you subject a protein to that intense heat, it's going to change the shape of the protein. Now, that is harmful to most of our proteins because our proteins like around 37 degrees. And so if you boil something, it's going to change the shape of the protein. And if you change the shape of the protein, it's not going to work well. But how do you like this? It was discovered in Yellowstone National Park in the hot springs that there's bacteria that actually live there. It's like, how can a bacteria, which has similar enzymes that we do, how can it survive? We always thought boiling water was going to be deadly to every living thing. So check this out. The bacteria that thrive in these hot springs, what they do in their cells, like here's a bacteria, what they do is they make DNA polymerase, but they make it, I find this to be one of the more interesting things, they make it misshapen. And so because they live in hot boiling water, the hot boiling water causes the protein to change shape into an effective conformation. So bacteria that live in the hot spring can only live in the hot spring because the heat changes the shape of the protein. So why am I going into this? Because we took that enzyme from the bacteria in Yellowstone. It's called amplitac DNA polymerase. TAC, it stands for thermoaquaticus, meaning that it can handle this kind of heat, and it amplifies DNA. So what are we doing with this? We are able to add that particular polymerase. Let me go back, sorry. We were able to add that polymerase to our test tube because it can handle the heat necessary in PCR. We also need this simple ion called magnesium. It's, the enzyme polymerase doesn't work unless it has magnesium. It's called a cofactor, and it's rather simple. Just that salt needs to be added to the test tube, and it's kind of curious because if certain enzymes need magnesium, one way to stop the enzyme from working is to not add the magnesium or try to put something into the test tube that gobbles up all the magnesium or chelates it, and so that's going to be something we're going to be considering later on. So it helps DNA polymerase, so it has to be there. Without it, DNA polymerase doesn't work properly. And so we also need DNTPs. This is a picture of just one of them. This is the adenine one. But there needs to be a DATP, like this one. And there needs to be DGTP, GTTP, and DCTP in the test tube. Or else, how can you make more DNA? You need the building blocks, which are the nucleotides. And now DNA polymerase can only grow them in the 3' direction. Just a little reminder there. And we need buffer, which is a chemical that we add that helps to maintain pH so that the enzyme works most efficiently at a particular pH. And so the steps of PCR is, number one is you need to open the molecule, so you need to denature it. And so the hot temperature will cause the molecule to open up, and how it does that is simply heat causes the molecule to pull apart. The only thing holding it together are these weak hydrogen bonds, and so tremendous heat opens them up. And then step 2 of the cycle is annealing. We take it down to a lower temperature so that the primers can attach to the target sequence. Here they are. This is called annealing. And then the third and final step of one phase is called extension. That's when DNA polymerase can start replicating DNA, and it attaches to the primers to do so. And so we use this machine called a thermal cycler to accomplish this. And what's interesting is the machine kind of helps us out because in our test tube, let's just review this for a second. In our test tube, we've got our template DNA. We have our, I'll use blue. We have our little primers that are going to attach here and here. We need our DNTPs. We need a little bit of our magnesium, and we need some buffer. And then, of course, we need our Taq polymerase. And this is it. So we put our redwood DNA in there, and we put our primers in. This is our primers. And here's our template DNA. And it's like, well, go for it. Well, again, you need hot temperature to open it up, and you need cool, and then you need warm to extend it. And so what this machine is that we put these test tubes over here inside this rack that's underneath this panel. And on the screen, we can actually program the machine to get hot, to cool, and to be warm, and then repeat. Hot, cool, warm, repeat. So we could do this for 5 cycles, 30 cycles. We could do it for 40 cycles. But can you imagine something that's been replicating 2 to the 40th time? We're going to be able to get millions and millions of copies of our DNA. This is called polymerase chain reaction. So open the molecule, attach, extend. And so typically a cycle parameter would be somewhere between 25 and 40 cycles. And the instrument that cycles between those 3 temperatures is a thermal cycler. And this is the one that we have. It's made by Applied Biosystems. And so finally, this is a little sort of sequence that goes through. Here's our redwood DNA. This is a little area called GAPDH that we're interested in. We get the molecule to open up using heat. There it is opening up. Then we get the primers to attach, and then the DNA polymerase extends in that area. So after 1 cycle, you have 2 molecules. And then after 2, you have 4. And then after 3 cycles, you have 8. And it continues and continues. And it moves on, and you get millions of copies of target DNA. So DNA is amplified, and polymerase chain reaction is able to amplify just the particular interesting sequence that you're curious about. It's quite fascinating. It's a great technique, and I'm looking forward to performing it with the class. So thank you for watching PCR.